Hi everyone, and welcome to Chapter 3, Take 2. This is the Chapter 3 lecture for microbiology, and it is all about the cell biology of bacteria and a little bit about archaea. So bacteria and archaea, what they have in common is they are both prokaryotes, meaning they don't have a membrane-bound nucleus. So the way that prokaryotes and eukaryotes differ is primarily their internal organizational structures. So eukaryotes have a lot of membrane-bound organelles, including a nucleus, which is where our DNA is found. In a prokaryotic cell, like a bacteria or archaea, the bacteria is sort of found just kind of floating around in the cytoplasm. They also don't have all these other awesome organelles that eukaryotes have. So these differences are pretty major in terms of the fact that a lot of pathogens of us, eukaryotes, are prokaryotic. And so finding the differences and understanding the subtle differences between the two cell types is actually really important for drug design. It helps us to design selectively toxic antibiotics that just kill prokaryotes but don't harm our cells as eukaryotes. So, parts of the prokaryotic cell. All bacteria have, there's a minimum number of structures that a bacteria must have. It must have a cell membrane, which is sort of an outer structure that holds everything in. It always has a cytoplasm, which is just an internal, watery, gel-like maybe environment of nutrients and chemicals that the cell needs to function. Floating around in that cytoplasm are these little orange dots here, which represent the ribosomes. Ribosomes are protein building machines. All organisms have genetic material that they can then pass on to their offspring. So the DNA in bacteria we'll see is different from that in eukaryotes in a few ways. One is that it's not found in the nucleus. It is kind of coiled up in sort of the center of the cell, call it the nucleoid. And it's also circular. Bacterial DNA is circular. And then lastly, they have some cytoskeleton proteins, some proteins that help give them shape. Most bacterial cells also contain a cell wall, which is an additional outer layer around the cell, and a glycocalyx, which is also another layer. So the green layer here represents the cell membrane or the plasma membrane or the cytoplasmic membrane. They all are the same thing. In yellow, that would represent a cell wall, and in orange here, the orange represents there's no line indicating it, but the orange would represent the glycocalyx, that surface coating. Some bacteria have additional bonus things, all right, including things for motility, like flagella or apillis, which they're pictured in green here, except I think this looks more like fimbriae to me. Usually pili are not so all over the place, but those are some different appendages that stick off the cell. They might have an extra membrane, a second phospholipid bilayer. They might have plasmids. Plasmids are extra bonus pieces of DNA, these little circles of DNA that bacteria can actually trade with each other, like Pokemon cards, I say. Some can form endospores, which is like a hard shell. They can form when conditions get tough. And those are really the main things I want to talk about. This is just another picture from your textbook that, we talk about how simple bacteria cells are compared to eukaryotes, but in this picture, they don't look that simple. They do have a lot of parts. So again, just to walk you through them, in yellow here, this would be the cell membrane or plasma membrane or cytoplasmic membrane, whichever you prefer, phospholipid bilayer, and then there's several other optional outer surface features. So most bacteria do actually have a cell wall, which is what is in magenta here. Some, the gram-negatives, actually have a second cell membrane, where did my cursor go, which is in the lighter pink here. Some have this protective protein layer called an S layer, and many have a carbohydrate layer, this outer sort of, whipped cream looking layer. is representative of carbohydrates sticking off of it, which helps bacteria stick to stuff. They may have these external appendages like fimbriae sticking off here. This is more like what a pilus looks like. It's like a tubular structure. Fimbriae are like little hairs. And here is a flagella, a tail-like structure, which is used for motility. Going inside, we have the blue jelly of the cytoplasm. All of those little purple dots represent ribosomes, our protein-making machines. This purple, stringy, wiry-looking stuff is the bacterial chromosome, the DNA, genetic material. And here is a plasmid right here, this little ring. And then you've got some very thin hair-like strands. Those represent the cytoplasm proteins that help give the shell its shape. And then these are rarer finds in bacteria. These are inclusion bodies or small membrane-bound compartments, which are kind of like organelles, but not, and sometimes they hold like packets of enzymes that the bacteria need. So bacteria can take on different shapes, and we have different names for those shapes. So a spherical shape is called a coccus. A rod shape is called a bacillus. If you're somewhere in between, you call it a coccobacillus. And there's a unique shape called vibrio, which is often described as comma-shaped. It's like a curved rod. So a vibrio that you might know of is Vibrio cholera, which is the causative agent of cholera, which is a really bad diarrheal disease. And there's also another form of vibrio that you're going to learn about in a TED Talk coming up about quorum sensing, which we'll talk about in this lecture as well. So multiple spherical, so single, coccus is single, cocci is plural, bacillus is single, bacilli is plural. So a single coccus, or you might have two cocci that are stuck together after the cell divides, and that's called a diplococcus. You can have bacteria that form in clusters. So we can talk about the shape of the bacteria. We can also talk about the arrangement of the bacteria. So those that form clusters are called staphylococci, and those that form straight chains are called streptococci. You can also have streptobacilli if there are bacilli in chains, or diplobacilli. If the cocci form these little series of four, so you see little like four packs everywhere, like four-leaf clovers, those are called tetrads. And if you have two tetrads together to make sort of like a cube structure, that's called a sarcina. And fun fact, I just read about a new sarcina bacteria that's causing really deadly, like 100% mortality, deadly disease in chimpanzees. If I forget where, I'm guessing in Africa, but I can't remember. And scientists are really worried about it because chimpanzees are very closely related to humans, and a lot of pathogens of humans also are pathogens for chimpanzees, which is why we use them in medical research. So there's a lot of fear of this bacteria crossing over to humans. It's called Sarcina troglodytes. Troglodytes is part of the Latin name for chimpanzee, and Sarcina because it forms these tetrad, or these sort of like double tetrad shaped cells. So there's a fun fact for you. Some bacteria can be shapeshifters to some extent. They can grow in different shapes depending on the conditions. So maybe like different temperatures. A bacteria might grow coccus as in a coccus shape at one temperature, but in a more of a bacillus or coccobacillus shape at another temperature. So microbes that can take on different shapes are called pleomorphic. All right, so now we're going to talk about the different parts of bacterial cells, and I'm going to start on the outside and work my way to the inside. So the external structures, starting with those that sort of stick off or are like appendages of the cell. So we have flagella, which can be used for movement, and there's a specialized kind of flagella called an axial filament. And then there are external structures that can be used for attachment purposes, and those are the fimbriae and the pili. So the flagellum looks something like this on a bacterium. It contains three parts. There's the basal body, the hook, and the filament. And basically, the hook is like an elbow, I don't know, like when you think of like a sink plumbing, okay? It's like this little elbow piece that connects to this pole of the basal body, and it spins around, and it spins that filament around. Ooh, I have a pipe cleaner, so I can be like, oh, just slapping myself in the face here. Probably too fast for the camera to see. Anyway, slow motion, flagella, going in a circle. It's like a propeller. Okay, so bacteria, what they can do is they can kind of spin it one way and spin it the other way, which I'll show you in a minute. So not all bacteria have a flagella, but those that do, they might have one. Some types of bacteria have one flagella. Some have one flagella coming out of either end, and some have a tuft of flagella coming out of one or both ends. So all three of these types are known as they have a polar arrangement because they have bacteria at one or both ends. So if they're at the ends, then they are polar bacteria. This one would be called monotrichous because it has one hair. Tricho means hair. Flagella is kind of like a hair. All right, so monotrichous. This kind is called lophotrichous. Lopho means a tuft. And this one is called amphitrichous, if a bacteria that has flagella at both ends, like an amphibian lives in both water and land. Amphibio means both. And then there's also bacteria that can have just flagella all the way around the perimeter, so we call them peritrichous bacteria, or peritrichous arrangement of flagella. So these flagella, remember, they rotate like propellers. All right, and so I'll put my face right here. So the bacteria move by basically twirling their flagella, and then they can stop and twirl it the other way. And when they do that, they sort of tumble around. So they spin one way and they start going in a straight line, and they start to get off course, so they spin it the other way to tumble around into a different direction. And so this process of moving is called run and tumble motility. And so the bacteria can use this motility to somewhat coordinate movement towards a substance or away from a substance. So the movement of bacteria is called chemotaxis, the movement toward or away from something. So in this picture on the right here, this bar, this gray bar, represents a gradient of, let's say it's a nutrient. So the dark black area represents a high concentration of nutrient in the environment over here, but low concentration over here. So a bacteria over here on the left is going to want to move towards that nutrient on the right. And so it will run and tumble and run and tumble and run and tumble, and it will overall end up running in the direction towards that nutrient. So we call that positive chemotaxis because you're moving towards something. But if this black area actually represented a toxin, something the bacteria wants to get away from, it would do the same thing but in the opposite direction. So we see this in bacteria and culture. If you have a culture of bacteria and you put a little drop of sugar on one end, the bacteria will collectively move through this run and tumble technique. And if you put a toxin on one end, they will move to the other side. So you can observe this movement of bacteria. If there is nothing special in the media, you don't add a gradient of any kind, the bacteria just kind of run around in the same place. They just kind of run in circles. So bacteria do drive themselves around. They just don't have much of a steering mechanism. So their steering is kind of random through these random tumbles and redirections. This is just another picture showing that run and tumble motility. Here is the path of a bacteria that is in an environment where there's no gradient of nutrient or toxins. They're just kind of running in place. And here's an example of one where they're in an environment where there is a nutrient gradient and they are trying to move towards it. It's, they take a very zigzag path. It's not a direct path, straight line, because of this run and tumble and lack of good steering, but they do eventually get there. So that's how flagella move in most bacteria. Spirochetes, the corkscrew-shaped bacteria, which I kind of skipped over and forgot to come back to. So here, I'll be over here. So we've got these two bacteria here, the spirillum and the spirochetes. So spirillum and spirochetes are kind of rare shapes of bacteria. Not a ton of bacteria have that shape. And the spirillum is kind of like, you can think of it as like zigzag or wavy, whereas the spirochete is corkscrew shaped. So the corkscrew shaped spirochetes are corkscrew shaped because their flagella is sort of internal. So it's underneath the cell wall, but outside the cell membrane. So it's sandwiched between those two layers, and it wraps around the long body of the spirochete. And so when the flagella moves and rotates, it rotates the whole body. And so you get that corkscrew motion. So that's kind of cool. So it's the same material that a flagella is on another bacteria, except that it's located inside. So we have a special name for it. It's called a periplasmic flagella because it's wrapped around the cytoplasmic membrane, or an axial filament because it goes all along. It's a filament that goes up and down the axis of the bacteria. All right, so those are motility structures, the different types of flagella. Now for some attachment structures. So one type are fimbriae. And fimbriae, I'll put myself over here, are represented by this dark shadow on the outside of the cell here, on each of these cells, that black shadow. I like to think of fimbriae as kind of like Velcro material on the surface of cells, because Velcro is sticky. It sticks to stuff, right? Kind of grabs it, like a bristly material. So this is like a very, very teeny tiny microscopic bristle material, and it does help bacteria stick to each other and stick to surfaces. If they decide they find a nice place they want to settle down. The pilus is a single tubular structure that they can basically shoot out and use to attach to other bacterial cells. And it's a tube, it's hollow, and they can actually transmit DNA and swap pieces of DNA, plasmids usually, through these tubes. And so it's a way that bacteria can share genetic information in a way that multicellular organisms cannot. So this, bacteria A can go up to bacteria B and basically inject it with some new genes. We can't do that. Humans can't do that. The only way I can get new DNA into my body is sex. And the DNA that comes in, if I get, fertilization of an egg, then I'm growing a new organism that has, different DNA from mine, but there's no way to get DNA from someone else into the DNA of my cells. I'm made up of millions and millions of cells. So at most, you could get the DNA into a couple of them, but that's not going to change my DNA. But if you're made of one cell, you're a bacteria, and you get some new DNA, that changes you. That changes your whole being. So bacteria can do that. They can just go around and share and swap DNA like Pokemon cards, just trade them out. And they do that using a pilus. So not all bacteria have a pilus, and not all bacteria can engage in trading of plasmids. But the ones that can pose a real problem for us clinically, because one thing that's very common on plasmids that we'll talk about are genes for antibiotic resistance. So it's how they can easily share that skill. So that's the primary purpose of the pilus. It's for that DNA swapping. That process, by the way, is called conjugation, kind of like a conjugal visit in prison. It's literally bacterial sex. There are different classes of pili. So pili can have additional functions. There's one class that's literally called the superpillis, and it can act as kind of like a flagella, a pillis, and a fimbriae. Like it can act for sticking, for conjugating, and for moving. So they can be multifunctional, but the main thing I want you to know about pili is that they're for conjugation, for swapping DNA. So those are all the different external appendages on the surface of bacteria. Now for some of the different surface coatings. So starting from the outside, moving in. So on the outside here in green, this represents a thick layer of carbohydrates, of polysaccharides on the surface, and this is called the Glycocalyx. And sometimes we more commonly refer to it as a bacterial capsule, and because it sort of coats the bacteria, kind of like, pills that have a smooth coating, have a capsule. So it gives bacteria, oftentimes makes the colonies look very smooth and glossy if the bacteria are encapsulated. The capsule sometimes is very slippery, and so the bacteria can, use it to evade the immune system. The immune system can start to swallow the bacteria, and it slips out of the capsule and the capsule gets swallowed. Or sometimes the capsule just tastes disgusting to the immune system. So we don't touch, you try to swallow it and then the bacterial cells, the immune cells are like, oh no, that tastes gross. That's totally an anthropomorphization of white blood cells. They don't actually have taste buds, but you'll get used to my analogies. So that's the Glycocalyx. It's sticky, helps bacteria stick, but also can help them evade the immune system. And the S layer is an optional layer underneath the Glycocalyx, which is made of proteins and is sort of like a chainmail, like how knights would wear armor and then they have the chainmail underneath the armor for just like some added protection there. So some bacteria have these things and some don't. So the Glycocalyx is particularly important for something called biofilm formation. And I bring this up, we'll continue talking about it throughout the course, but it's particularly important clinically for pathogens. So we tend to think of bacteria as these individual cells, right? We're talking about these individual bacterial cells. But bacteria behave very differently when they're alone or in low density populations versus when there's a lot of them around. I mean, you can kind of think of them, we have insects that we call social insects, like ants and bees, who by themselves, can't do much, but together they can create these amazing structures of like a beehive or an anthill, right? Bacteria can do the same. Bacteria can actually work together and talk and communicate with each other, which is really fascinating and a relatively recent discovery, I don't know, maybe the 1990s. So this is called, this use of chemicals for bacteria to talk to each other and sense the size of their population is called quorum sensing. So all bacteria secrete these little molecules that when they build up in high concentration, when there's a lot of bacteria around, it triggers certain genes to turn on. So in this single bacteria here, when there's not a lot around, it's light blue, right? But then when there's a lot of them around, they turn bright blue, right? They start producing different proteins. One of those things that they can start producing is a Glycocalyx. So when you have planktonic bacteria, those are just swimming around, looking for a place to settle down and build a home, right? When they find the nice spot, they might start secreting their Glycocalyx then, and then they're nice and sticky, they stick, they keep growing, and they form this, dense community, and they can start talking to and recruiting other microbes to that spot, and you end up with a thick film of microbes that's oftentimes multi-species. Lots of different species of microbes there. These films are particularly difficult to treat, to get rid of, They're somewhat impervious to antibiotics because they're actually physically dense and thick, so the antibiotics can't always get in. People who get like chronic internal infections are sometimes due to biofilms, and it can be very difficult to treat them. So studying biofilm formation and the type of chemical chatter that goes on between bacteria and tells them to turn on things like toxins or turn on certain genes that make them cause disease is a really important thing to understand in order to prevent disease or treat disease. So you're going to be watching a TED Talk about like one of the bigwigs in quorum sensing, but I've watched a couple of others that I didn't assign where they talk about drugs that they are designing to block this quorum sensing feature so that the bacteria can't turn on these toxin genes. And also another TED Talk where they use tests to detect these molecules in order to sort of be able to spy on and listen to bacteria that are planning an attack. And the chemical tests for these bacteria are more sensitive than the chemical tests for the bacteria themselves. So you can basically hear the bacteria before you can see them in a way. And it can help to, again, early intervention for treatment of disease, potentially. So these molecules have potential therapeutic value and also diagnostic value to prevent biofilms and to treat biofilms, which cause a lot of disease. All right, so some other structures on the outside of the cell are the cell wall and the cell membrane, and collectively we refer to these things as the cell envelope. You know, an envelope is something you put a letter in and it covers up, wraps around the letter. All right, so these are the layers that just wrap around the inner contents of the cell. So all bacterial cells have a cell membrane or plasma membrane or cytoplasmic membrane, a membrane, okay? And this is made up of phospholipids, as we'll see. And then they all, well, not all of them, but most of them have a cell wall, which is made primarily of something called peptidoglycan, which is like a carbohydrate with little protein elements mixed in. Okay, so gram-negative cells have a very thin cell wall or peptidoglycan layer, and gram-positives have a very thick cell wall or peptidoglycan layer. And gram-negatives, because they have such a thin cell wall, have a second membrane or an outer membrane on top of it for additional sort of support. So gram-positive and gram-negative, they get those names actually from a staining technique that was developed by a man with the last name Gram. Gram-positive cells stain blue, gram-negative cells stain pink, and I'm going to go through that procedure in a minute, but this is just another molecular view of the differences in these cell wall structures. So gram-positives have this very thick cell wall, this thick peptidoglycan layer. Over their cell membrane, here's this phospholipid bilayer of the cell membrane, versus gram-negatives, which have a very thin peptidoglycan layer, which is here in magenta, sandwiched between two phospholipid membranes. They also have different surface molecules. So gram-positives have lipodocoic acid as a fatty acid that sticks off the membrane, and the classic one of gram-negatives, the important one, is called lipopolysaccharide. So the phospholipids in their membranes that have these little carbohydrates sticking off of them are called LPS or lipopolysaccharide, and they happen to induce an inflammatory response in our bodies with our immune system. They induce fever and inflammation, and so LPS is also commonly known as endotoxin. So endotoxin, LPS are synonyms. All right, they're called endotoxin because they're found in the membrane and they're toxic to us. So let's look a little bit more closely at the cell wall structure. Cell walls are made up of peptidoglycan. This is an important molecule to know. It's part peptide, little like short peptides here in yellow, and part carbohydrate. That's these hexagon, the pink and green hexagon here. These are polysaccharides made of 2 different monosaccharides in repeat, and that are linked together by these little short amino acid strands, these peptides. So it's both part protein, part carbohydrate, and it makes up this important structural material like a chain link fence around the cell membrane. And it's important, the cell wall is important for bacteria because bacteria, again, they're single cell, and cell membranes are semi-permeable. So that means water can freely move in and out of the cell. And if too much water moves into the cell starts to swell and can rupture if there's too much osmotic pressure. And that's what the cell wall is primarily for. It really helps the cell not rupture. It helps it to withstand that osmotic pressure when it goes into different, concentration environments. So that's why most bacterial cells do have a cell wall, because the cell wall is important for maintaining integrity when there's a lot of osmotic pressure. So gram-positive and gram-negative cells get their name from this staining procedure. And I'm actually going to walk you through the staining procedure because it's pretty important. It's pretty standard. It's done in like every diagnostic hospital microbiology lab. And understanding it also means that you actually understand the differences between the gram-positive and gram-negative cell walls and cell envelopes. So the gram staining technique results in 2 different colors depending on whether a cell is gram-positive or gram-negative. And it is 4 steps, 2 different dyes. The first dye is called crystal violet, and crystal violet is like a deep blue color, like a dark violet or blue. And if you add this to the cells and then you wash it off with water, the water won't wash it all away. It'll wash it away from the environment, but it'll stick to all the cells and make them all purple. So you could just stain everything. That would be a simple stain, staining everything with crystal violet and then just looking at it, okay? Because both the crystal violet goes into that peptidoglycan cell wall layer of both types of cells and even into the outer membrane a little and sticks there, okay? The second step is called the mordant, all right? Iodine is a mordant. It causes the crystal violet to kind of clump together. So here you can see the dye molecules are kind of like spread apart, and here they clump together. All right, they don't clump particularly well in gram-negative because the peptidoglycan layer is so thin. There's such a small amount of dye in there, they don't get too clumped together, but they do still clump. Then the next step is called decolorization, and you add an alcohol. An alcohol dissolves membranes. So it doesn't dissolve the inner membrane, but it does dissolve the outer membrane of the gram-negative cells. And with it, it washes away the crystal violet. It doesn't wash away the crystal violet in the gram-positive cells because since you added the iodine, it caused the stain to clump together, and now those clumps are really trapped in that peptidoglycan layer. If you forgot to do the iodine, then the dye would wash away from both of them. Both types of cells would be decolorized. So the iodine is important to make it stick in the gram-positives. So you could stop here if you wanted, and you would definitely be able to pick out gram-positive cells. The gram-negative cells would be clear because you would have washed away all the stain. And clear cells are really hard to see under the microscope because they're have the same refractive index as the glass. And so to make them more easily visible, we add a second stain, and this one is pink. It's called safranin. It's like a bright pink dye. And so all the cells will take up the safranin. Those that already have crystal violet, well, the crystal violet and the safranin, it's a much darker color than the safranin. So crystal violet by itself is maybe like a dark blue, and when it's after this last step, it becomes maybe like a bluish purple, like a dark purple blue. So gram-positive cells appear like dark purple blue, and the gram-negative cells appear pink or red. So most cells do fall into that gram-positive or gram-negative category, but there are other cells that have atypical cell walls or no cell walls. All right, mycoplasmas are one of those. They don't have a cell wall at all, but they do have very stable membranes. There's a biotech company in Saranac Lake that actually does mycoplasma testing. So mycoplasma bacteria are not harmful, but they are common contaminants in labs that do tissue culture. And there's tiny little bacteria and they don't really cause any problems, but when you're doing research, it's really important that the cells that you're using to study are not contaminated, because who knows how the contamination might be affecting your results. And so their job is to test for mycoplasma contamination and clean it if there is any. Mycobacterium is different than mycoplasma. Mycobacterium do have a cell wall, but it's different. It has different acids in it than gram-positives. And so there's a different staining procedure we use called acid-fast staining, which kind of looks like gram staining, because it also uses a blue and pink dye. But a famous mycobacterium, two of them actually, Mycobacterium leprae, I don't remember, it causes leprosy, and Mycobacterium tuberculosis, which causes tuberculosis. So they're important too. And then there are the Archaea, which many do have cell walls, but their cell wall structures are very different than the gram-positive and gram-negative cell walls. None of the Archaea are known pathogens, so we don't spend a lot of time on them in this class, but I'll touch on them a little bit in this chapter, just so you don't forget they exist. So then the last thing that wraps around the cell besides the cell wall is that cell membrane. Membranes are always made up of phospholipid bilayers, which is sort of the basis of the material of the cell membrane. And then there are proteins often embedded in it. So you can have proteins that act as signaling molecules, ones that have carbohydrates attached to them that act as receptors, and ones that are shaped like channels and act in transporting things in and out of the membrane. So membranes are what we call selectively permeable. Some things can go in through the cracks in the phospholipids, but but those are only like really small things. Water, gases like oxygen and nitrogen, but mostly, like even glucose, which is a relatively small molecule, has to go through a transport channel. So yeah. So some of the functional differences between gram-negative and gram-positive, we know there's structural differences, what the differences in the structures are here and how they stain differently. But some of the things that cause how these contribute to functional differences. So gram-negatives, having a second membrane there, membranes control what goes in and out of the cells. So having two membranes means you have sort of double barrier of things going in and out of the cells. So in the case of treating bacteria with antibiotics or drug therapy, a lot of times gram-negatives are more resistant just because they sometimes don't take up the drug. Their two membranes end up sort of blocking it and not allowing the drug in, and the drug doesn't work if it doesn't get in to poison the cell. But gram-negatives are also more sensitive to alcohol, sterilizing with alcohol, because of that outer membrane is very easily degradable with alcohol. Membranes degrade alcohol, and so they don't live as well on surfaces because they're easier to sterilize. Whereas gram-positives might survive better on surfaces or survive alcohol sterilization better, but they're slightly more sensitive to antibiotics because they don't have a second membrane to block that uptake. Just some trends. It's not always the case. All right, so now we've talked about all the external structures. Now for the internal components of a bacterial cell. So cells are full of fluid. That fluid is called the cytoplasm. and it has nutrients and chemicals and stuff dissolved in it. Bacteria also always have DNA. They need genetic material. So bacterial chromosomes are circular, which is unique and different from eukaryotes. Our chromosomes are in pieces, like linear pieces. So eukaryotic chromosomes are linear. Bacterial chromosomes are circular. Bacteria also can carry extra pieces of DNA, of circular DNA, called plasmids, and these are the pieces of DNA that they can swap, like Pokemon cards, using a pilus. These plasmids contain, I like to think of them as containing bonus genes. So in the chromosome are all of the genes that the bacteria needs to live and survive. And then the plasmids might contain bonus genes for like toxins or antibiotic resistance. If the plasmids contain useless genes that the bacteria doesn't need, it'll often just kick them out and get rid of them. But if they are useful, then they hang on to them and share them with their friends. Another thing inside of bacteria and all cells are ribosomes. Ribosomes are the machines within the cells that basically make proteins from our genetic coding. So our DNA codes the protein, has the code for it, and then we use mRNA, we use RNA to make copies of that code, bring it out to the ribosome, and the ribosome translates it into protein. We'll talk way more about that in chapter 8. So prokaryotes, bacteria, and eukaryotes, fungi and parasites, and us, all have ribosomes, and they're pretty darn similar, but they're a little bit different, and that difference is enough to be meaningful. So bacteria have, so ribosomes in both types of cells, prokaryotes and eukaryotes, have two subunits that combine and come together to make the functional ribosome. So here's the two tertiary structures of these. These are proteins. Ribosomes are made of protein and a special kind of RNA called rRNA, all lumped together, so mixed together. So these two tertiary structures come together, make a quaternary structure, and then it can do its job. So 50S and 30S, the S is a unit of measuring protein size. Okay, so prokaryotic ribosomes are of the 70S variety, and eukaryotic ribosomes are 80S, so they're a little bit bigger than these prokaryotic ribosomes, which seems potentially insignificant, a minor detail, but ribosomes are pretty darn important for a cell to be able to function. Without them, the cell will die. And so since there is an actual difference between prokaryotic ribosomes and eukaryotic ribosomes, they can serve as an excellent target for drugs, for antibiotics, because you can find ones that selectively bind to the prokaryotic ribosomes and not the eukaryotic ribosomes, so the bacteria die and we don't. Another subcellular structure, some bacteria, these are much more rare, have things called inclusion bodies, which are little packets maybe surrounded by membranes that they use to store things, like maybe store enzymes, or they might store gas for buoyancy if they're marine bacteria. There are some that are known to store iron for magnetic purposes, that they can they're basically like little bacterial compasses, and they can sense gravity or something through magnetic poles of Earth, and their iron storage compartments. So there's a lot of crazy, cool bacteria out there that have cool extra things going on for them. And then the last thing that's inside of bacteria, the last important protein that's found in the cytoplasm, is the cytoskeleton proteins. And these are, our skeletons are made of bones. Bacterial skeletons are made of proteins, and they're not nearly as specifically structured as our skeletons are, but it does help them. The reason that a bacterial cell is a coccus or a bacillus, that shape is held in place by those cytoplasm filaments. And another thing that these cytoskeleton filaments are important for is cell division. When bacteria divide, And this is a picture from a Nature Reviews paper that was actually by a postdoc who was my TA in college, who worked in a lab. One of my microbiology professor actually in college, she studied these cytoplasmic proteins, particularly her favorite one was this one called FITC. And it's really important for causing that cleavage of the cell when it divides. And so the idea is there's potential therapeutic value if you could find an antibiotic that binds to FITC or somehow disables the cell's ability to cleave, then it would inhibit bacteria division, or they would divide, and instead of having two bacterial cells, you have like these Siamese twin cells that are stuck together and can't function very well. So that's another important job of the cytoskeleton proteins is in cell division. And then some bacteria have abilities to form endospores, which are like these really hardy shells that can help them survive in really poor environmental conditions. So like if it's really dry or really hot, or there's not a lot of nutrients around, the bacteria can basically like tuck into this hard shell. And then when they're placed in an environment that does have plenty of nutrients and water and whatnot, they grow. So they're kind of like seeds, like plants produce seeds that then, can stay dormant for a while, and then you put them in a favorable environment with water and dirt, and they germinate and start to grow. So some bacteria you should know that form endospores. Bacillus anthracis is the causative agent of anthrax. You may recall, I think it was in 2000, but don't quote me on that, there was some bioterrorism efforts using anthrax spores via the mail. And I think several people at the Pentagon actually received letters with this powdery white substance in it that was anthrax spores. And they can be breathed in, and then they can germinate and cause a respiratory infection. And a few people did get sick. Clostridium species are all spore formers, endospore formers. So one of these, it causes tetanus. So think tetanus, you think of getting, a cut or getting a puncture wound from some rusty equipment. So rusty metal is not a great site for bacteria to grow and thrive. There's not a lot of nutrients there for them. So they're not typically covered in bacteria, but they might be covered in bacterial spores of Clostridium. And then when it punctures into fresh living tissue, the endospores germinate and start to grow and cause an infection. So the life cycle of a sporulating bacteria looks like this. So when conditions are good, they're just dividing and growing and dividing and growing. That's called the vegetative cycle. But when conditions are poor, like it's really hot, really dry, or really cold, all right, then it'll activate this spore formation life cycle. And so it'll start to sporulate, so it'll divide. The cell will divide, but instead of splitting into two different cells, the daughter cell ends up getting encapsulated by several layers of tough protein. All right, and then the mother cell basically dies and disintegrates, and what's left is this spore, which can live for ever, potentially. I forgot to mention on this slide, but they've actually found a spore or some spore, an endospore, a bacterial endospore in like a polar ice cap in like Antarctica, deep in the layer of ice, that was dated to be 250 million years old. I can't recall if they were able to germinate it or not, but certainly thousands of years. In fact, one of the hypothetical fears with global warming is that, there are bacteria that could have been pathogens, ancient pathogens that, sporulated in the ice caps or in like the tundras. And now that global warming is melting these things, then these endospores that have been preserved for centuries or millennia may start germinating and return as pathogens. That's a real concern. So that's how sporulation works. This is a picture up here on the right of some, I think, Clostridium cells. So this is a vegetative cell right here. And these are cells that are sporulating. So you can see the tail part there, and then here's the endospore that's forming. So they look kind of like lollipops. Archaea, I said we won't talk a lot about them because they don't tend to be pathogenic. They don't cause disease, but they are really cool. So, if you're interested in learning more about them, go for it. They tend to be extremophiles. They're called Archaea because they're archaic, they're old, and they tend to live in environments that are similar to early primordial Earth. Funny enough, we, for a long time, they don't have a nucleus, so we grouped them with bacteria, thinking that bacteria and Archaea were kind of the same, that Archaea were just a type of bacteria. So we had prokaryotes and eukaryotes, but it became clear, especially once we had genetic tools to look at genetics, that Archaea are their own domain. So they're not bacteria, they're not eukaryotes. There's something in between, maybe. Although their cell structure, the fact that they don't have a nucleus, makes them look more closely related to prokaryotes. If you look at them genetically and at some of their cell structures, they actually are much more closely related to eukaryotes. So they're kind of like maybe like a missing link in cell lineage. So they're kind of interesting from an evolutionary standpoint to study. And they're interesting in the types of environments they live in because they tend to be extremophiles. They love extreme conditions. So there's Archaea that are psychrophiles. They grow in the cold. They live in like polar ice caps. There are ones that are thermophiles or even hyperthermophiles in like hot environments and live in like tar pits and thermal vents and geysers and stuff. And there's ones that can tolerate really high salt concentrations and live in the ocean or even live in like the Dead Sea. And we call those halophiles. So Archaea are cool because they tend to be extremophiles and live in conditions that we think of as not being supportive of life. So bacteria can be classified taxonomically, kind of like we do for organisms. And the sort of field guide for classifying bacteria is called Bergey's Manual of Systematic Biology or Bacteriology. And Bergey divided everything based sort of phenotypically. What type of cell membrane do they have? Do they have flagella? Do they metabolize certain sugars? And so you can make these diagrams that you follow, like a flow chart, called a dichotomous key, that you can use just like you can do for, trees. Like, does it have points on the leaves? How many, stems, whatever. Like, so you can figure out what type of tree you have. You can do the same with bacteria. But in recent years, with the development of DNA technologies, the bacterial classification system has kind of changed, and we found that some bacteria that look a lot alike are actually not very closely related. Bacteria, although they, all are these single-celled organisms, are highly genetically diverse. So two bacteria could be as different as we are from an ant or something like that. So they can be pretty different, even though they all kind of look the same to us. So the four main divisions in systematic bacteriology are Gracilicutes, Firmicutes, Tenericutes, and Mendosicutes. And basically they all have different cell wall types. So Gracilicutes are where the gram-negatives are classified. Firmicutes are where the gram-positives are. Tenericutes are those that lack a cell wall, like mycoplasma, and Mendosicutes are ones that are archaea. So when it comes to bacterial species, defining a species, it's a little bit different than in higher level organisms. So in animals, species are defined as two things that can't procreate, or if they do procreate, their offspring are infertile. So for example, donkeys and horses are two different species. You can mate a donkey and a horse and you get a mule, but mules are infertile and they can't go on to reproduce. So we say that that donkeys and horses are two different species. Bacteria can mate all over the place, right? They've got those pili, so they can actually exchange DNA with other species. And bacteria are also asexual, so they procreate on their own. They don't have to procreate through mating. So we can't use that same definition of species. So the definition of species in bacteria is a little bit more nebulous, but has to do with genetic similarity. So they usually share traits, but they have to share at least 70 to 80% of their genes, which is still like not a ton. I mean, humans and chimpanzees are like, I think chimpanzees are like 98% the same genetics as humans, and we're a different species. So bacteria have a lot of genetic variability, and we can still say that they're the same species. So a classic example is E. coli. So E. coli is a species of bacteria. The full name is Escherichia coli. And there's Escherichia coli living in all of your guts right now. And it's perfectly normal. It's perfectly healthy. I call it vanilla E. coli. It's just plain normal E. coli. All right. But there's another strain of E. coli. Well, there's several strains of E. coli that are pathogenic, that cause disease. And the first one that really came onto the scene was in the 1990s in a foodborne outbreak. And the strain is called E. coli 0157H7. And it causes really bad diarrhea, and it can also lead to kidney infections. So it causes foodborne illness. Whenever you hear about E. coli poisoning in a pool or in ground beef or in spinach, we're talking usually about E. coli 0157H7. There's a couple of other pathogenic strains as well. And the difference really between them and our normal bacteria, our normal E. coli, is that they picked up along the way, somewhere actually probably evolved in cattle stomachs. Some E. coli bacteria in a cow's stomach ended up picking up some new genes and now can make a toxin, and that causes us to become very sick when we ingest it. So even bacteria that we usually think of as bad, E. coli, we're used to hearing it as a pathogen, as a disease. That's just one of the members of the E. coli family, of the subspecies or strains of E. coli. So not all strains of E. coli are bad. Some are harmless and maybe even good for us. But even those strains aren't always good, because even our normal E. coli can make us sick if it gets into the wrong part of the body. Very classic example is urinary tract infections. If you wipe the wrong direction and you get a little bit of fecal material in the pee hole, your urethral meatus, it can start a urinary tract infection. The most common cause of urinary tract infections is E. coli from your gut that got into your urinary tract. So even bacteria that are normal and healthy in one part of the body can cause disease in another part of the body. So we can't really call any bacteria good or bad, but I will still do that anyway. So lots of bacteria are like this, that there's different strains. Some might be pathogenic, some might not be, or they might cause different forms of disease, etc. And that is where I will stop.